you. Okay, and then somebody else, Tiffany. Let's see if I can find Tiffany first. Okay. Yeah, so it's easier if someone can. So Kate, Cooper, I just gave you permission too, but it's easier if one person can just record it and then share it. You guys can send it to me, and then I can just put it on Canvas to share with everybody else. Would that work? Okay. All right. So I gave I gave both Cooper and Kate permission to record. I think it will be safer to have two people, and then if one person end up having technical issue, at least we have another backup. Let's try that and then see how it goes. So go back to the Kahoot question. So this one says cholesterol serve several essential functions in the membrane. Which of the following is not influenced by cholesterol? And the middle two, so membrane fluidity and also the rigidity. So these are definitely the main function of cholesterol. So those are definitely part of cholesterol's role. By inserting itself into the membrane, it's also kind of affecting the permeability of the membrane. So the correct answer we're looking for is actually the membrane thickness, because the membrane thickness, if you guys remember one of the first slides, we said the membrane is about 5 to 10 nanometer, and that's totally decided by the two layer of the phospholipid. Okay. Yeah, so there is for Kahoot, it is graded not based on accuracy, it's just based on participation. And of course, you want to get as many, correct ones as possible. But you're not graded based on accuracy. So let's see. So we have Bahar, Anina, and Kylie, Jamie, and Helen on the leaderboard. Okay. 23 is not bad. So this one is a little bit tricky, and it says which component can be found either on the surface or as part of the membrane. The correct answer we're looking for is protein. Some of them are spanning the membrane. Other ones are kind of loosely associated. If you look at carbohydrates, so they're only on the outside. So they're not really the integral part of the membrane. Phospholipid, definitely the integral part. And cholesterol is only on the inside and they're not on the surface. So that's what make up the membrane. And of course, one of the main property of the membrane that makes them important for their function is this property be called selective permeable. Basically, they are selective in what they allow to go into the cell and what they allow to come outside of the cell. Kind of as a side note, a lot of times, so we keep talking about the bilayer, and then people might be under impression that the two layers are actually identical. In reality, most membranes are actually asymmetrical. So if you think about it, the ones that are facing the extracellular matrix, a lot of them are being modified with sugar molecules, for example. And the ones that are facing the cytosolic part are actually not. So membranes in general are asymmetric. And the permeability, of course, a lot of it kind of depends on the property of the membrane. So remember, it is made of lipids, and that kind of determines who can go in and who can go out kind of more easily. Okay. So generally speaking, when it comes to think about what kind of molecules can easily cross the membrane versus not, there are two properties. So, one is size. So in general, small molecules have a much easier time to cross the membrane than large molecules. And then, of course, the other property is polarity. So because membrane themselves are made of nonpolar molecules, they're lipids. So things that are nonpolar have a much easier time to cross the membrane than things that are polar. Of course, it's not absolute. So, water, for example, it carries partial charge, but water is small enough that it can still cross the membrane on its own. Although in cells, most of the time that's too slow by diffusion, so you actually don't rely on that. We do have aquaporin to help water cross faster. And the charge is definitely, so if you carry a lot of charge, so think about ions, sodium chloride, for example, they are really small, but because they carry a lot of electric charge, they won't be able to cross the membrane by themselves. So they definitely have to rely on transport protein for them. And then, of course, large molecules like proteins or polysaccharide and stuff, they won't be able to cross the membrane. So we have things are changing. So we have Bahar, Kylie, Jamie, Helen, and Bill on the leaderboard. So this is another visual for kind of judging what kind of molecules can cross the membrane or, have an easier time crossing the membrane than others. So small nonpolar molecules like oxygen, for example, CO2, they can totally diffuse across the membrane. And even steroid hormones, remember they're lipid-based and they're small enough and they can actually cross the membrane on their own. Small uncharged but polar molecules like water, they can diffuse across the membrane, but they're actually doing it very slowly. Ethanol and glycerol are small enough, so even though they are a little bit polar, they can still cross the membrane. Large polar molecules like glucose and amino acid, almost no. You can see the size of the arrow; it's very, very thin. So things like amino acid and glucose are considered nutrients for the cell. And most of the time, cells actually do have carrier proteins to basically carry them through the membrane. And then ions are just way too charged, so they won't be able to cross the membrane at all. And you have to use channel or transporter to get them through the membrane. So let's talk about the transport process. So the simplest transport, of course, is diffusion. So diffusion is basically molecules are moving around all the time. So they're colliding with each other, and they do have a tendency to go from a high concentration to a low concentration. So if across the membrane you have a concentration gradient, the molecule will always move from the high concentration to low concentration gradient until they will reach this point called equilibrium. And when they reach the equilibrium, it doesn't mean that molecules are not moving. It just means that the moving, going in and going out the rate of the movement is pretty much equal, so they cancel each other, so there's no net movement of the molecule. But some molecules are still going in and some other molecules are still going out. So that is for molecules that can cross the membrane on their own. So for molecules that cannot cross the membrane on their own, if you look at this picture over here, for example, so that hexagon represent, let's say that's glucose, right? Remember, it's a 6-carbon molecule. So it cannot cross the membrane, and then we actually need the help of these carrier protein to help them cross the membrane. And because it's also going from high concentration, high glucose concentration to low glucose concentration, it's called facilitated diffusion. Diffusion basically means that they're going from high to low concentration. Because it's energetically favorable, because they're going from high to low concentration, it doesn't need you to input a lot of energy in there. So it doesn't really need ATP, and it is a passive transport. So basically that's, set it apart from active transport where you have to put in energy. So when it comes to channel, especially for this simulation, which a lot of you guys have done, some channels are actually just not gated, they're open all the time, but most channels are actually regulated. And that's because the cells will actually adjust its need, so it may not need glucose all the time, or whatever it's transporting all the time. And so that's called gated. And then there's voltage gated, there's mechanical gated, but basically a change in the membrane potential can actually trigger a channel to open and then allow whatever their transport to cross the membrane. So the carrier protein in general, basically what happens is they can change their conformation. So, this again is a picture of a glucose transporter. So it can bind to glucose. There's higher glucose concentration on the outside, so it binds to glucose and then it changes the conformation. And then on the other side of the membrane, it can release glucose into the cytoplasmic side. So these carrier proteins are usually very specific to whatever they're transporting. So if it's a glucose transporter, it probably won't bind to another sugar like fructose, even though, strictly speaking, fructose is very similar to glucose, but a lot of these are very, very specific. Okay, this one says which molecule will pass through the membrane by just simple diffusion. And if you look at the list, it actually should be relatively easy to pick it up because protein is way too large to cross the membrane by diffusion, so it never crossed the membrane by diffusion. Starch is way too large to cross the membrane by diffusion. Sodium ion is way too charged to cross the membrane by diffusion. So basically the only thing that can cross the membrane by diffusion out of this list is oxygen molecule. And we have Bahar, Jamie, Vil, Kylie, and Helen on the leaderboard. Okay, so that's diffusion and facilitated diffusion. Let's talk about osmosis. And traditionally, and I think in this class too, there has been a lot of confusion about osmosis. So let's talk about it. Spend a little bit of time talking about it. So osmosis is literally the diffusion of water. So let's say you have a semi-permeable membrane that separates compartments, and then you have a solute that's trapped on one side. So normally if that solute is small enough to cross the membrane, it will try to go through the membrane, and then it's, eventually it will establish equilibrium. But because the solute is not able to cross the membrane on its own, basically what happens, so we call these non-penetrating solute because it cannot cross the membrane. What happens is the water is actually going to try to cross the membrane to try to even out this concentration gradient. So if you have a 10 molar, glucose solution or sucrose solution, whatever, and then the other side is just pure water. Water is actually going to try to cross the membrane to dilute it, so that the concentration gradient between the two is not as steep. So that part is actually not too bad. But the part that does get confusing is actually, so if you think about it so we always talk about the solution from the solute point of view. So, when we talk about 10 molar, sucrose, it's literally 10 molar sucrose dissolved in water, right? But when it comes to osmosis, it's actually easier to think about it from the water point of view. So if you look at the two solutions, the 10 molar, sucrose solution has some water, but it also has a lot of sucrose. On the other side, it's pure water. So that side is made of 100% is water. So the water concentration on the left side is actually a lot higher than the water concentration on the right side. So the water is literally diffusing down its concentration gradient. So it's going from high water concentration to low water concentration. And to predict which direction water is moving, there is a saying that's called water follows salt. So the salt over here does not necessarily mean sodium chloride. It means, any non-penetrating solute. So basically, if you know that on the right side, the solute concentration is higher, that's the direction water is going to move to. So water is going to, try to go from left to right to even out the concentration gradient. Okay. Okay, so we have 32 answers, and I don't know, so on paper, there should be 34 of you guys. So I don't know if someone's having technical difficulties, not participating. But yeah, so the correct answer we're looking for over here is isotonic. So 29 is pretty good. I think our record was 30 today. So for red blood cell, so yeah, Amy, you have techno. I don't know. I mean, anybody else, if you guys have tip for Amy, go ahead, let her know. I don't know if reloading it will help. But yeah, sometimes Kahoot does drop you, and people do have difficulty. So for red blood cell, animal cells, remember we don't have a cell wall, so that means our cells are more vulnerable. And guys, don't use nickname. If it doesn't allow you to sign in with your full name, you can just change it, use abbreviation. Because if you use nickname, I don't know if it's you, right? So when I download the report and it says, something about kangaroo, I have no idea who that kangaroo is. Sometimes Kahoot generate really cute nicknames, but for for the purpose of participation, that actually doesn't serve you very well. So yeah, so let's talk about tonicity. So I think in the pre-lab quizzes, there are a few people who get confused about the tonicity. So tonicity literally means the relative solute concentration. So early on we thought, so let's say you put your red blood cell into a beaker, and an isotonic solution just literally means that the solute concentration in the beaker is equivalent to the solute concentration inside the red blood cell. And basically what that means is, because remember, water always follows salt. So some water will move into the red blood cell, other ones will move out of the red blood cell. But in general, there's no water net movement. And then the red blood cell is actually usually pretty heavy in isotonic solution. If the beaker contains like pure water, for example, so the concentration, the solute concentration inside the red blood cell is much higher than outside, which is pure water. So that pure water is called hypotonic solution. And then of course, water will follow salt. So more water will move into the red blood cell. And then eventually the pressure gets to be so much that the cell will actually burst. So that's called lysis. And literally it will take, if you do this experiment in the lab, it will literally take seconds for all the red blood cells to just disappear because the membrane just cannot sustain that kind of pressure. And then if you put your red blood cell into like a 30% sodium chloride, for example, so the salt concentration on the outside is much higher than what's in the inside. So that's called hypertonic. Then of course the water will move out of the red blood cell, and then the cell will actually shrivel. So it basically become very dehydrated, and that phenomenon is called crenation. And I don't really know how. So let me see. So Anina was asking something about, in terms of the extracellular fluid has more water than the solute concentration. Well, I mean, so if you go into physiology, and I think I do have one of the slides to show you. So there's the inside the cell, so you have the cytosol, which is water-based, and then outside of the cell you have the extracellular fluid, which is kind of water-based, and then of course, a lot of times things will also move into the blood vessel. So you also have the blood volume. So you are constantly comparing the solute concentration in two compartments. So you might be comparing it between the extracellular fluid versus the cytoplasm, or between the blood and also the extracellular fluid. So that's actually why we're learning this, because it's totally relevant in terms of physiology. Okay, so plant cells, unlike animal cells, actually they do have cell wall, and that means if too much water gets into it, the cell membrane will actually be pushed against the cell wall, and then basically, the plant cells actually really prefer to be in hypotonic, and that is because when the plant cells are in isotonic solution, so there is no net water movement, right? So some water moves in, some water move out. This kind of condition is called flaccid for plant cells. So the membrane is not exactly pushed against the cell wall. The cells are okay. They're not dying or anything, but they're not in their happiest state. In hypotonic, when you have a lot more water moving in there, basically that will fill their central vacuum, and then it will actually push the membrane against the cell wall, and this is called turgid, and this is actually when the plant cells are the happiest. And then of course in hypertonic, they're losing so much water, so they also become very dehydrated. If you look under the microscope, the membrane will actually peel off from the cell wall. The central vacuum doesn't contain a lot of the water, so that state is called plasmolyzed. And plant cells like it in hypotonic because it actually gives them target pressure, and they actually rely on, especially for non-woody plant, they actually rely on that target pressure to support the plant. So if you go on holiday for, for a month and you forgot to ask somebody to water your plants, you come home and this is kind of what your plants look like. So they become wilted. And that's because they have lost so much water and they have lost the target pressure and stuff. So fortunately, most of this, is actually reversible. So if you start watering your plants, they will actually look healthy again. So plant cells do have different requirement, and this is kind of on the side, but I have the slide, so I thought I might as well mention it. So remember, so animal cells and also a lot of the protozoa, like things like paramecium, for example, over here, and like amoeba, for example. So paramecium and amoeba actually live in fresh water. So that means that water has got to be constantly going into the cell, right? But how come they don't go bust? And that's because they actually have an organelle that's called contractile vacuole. So the contractile vacuole can actually regulate water, and it can collect all the excess water and then try to pump it out. And then that way the organism is not under too much osmotic pressure. And then there are other things that live in marine environment, like the jellyfish, for example. They actually have a mechanism to match their solute concentration inside to the solute concentration in the seawater. And then that way they actually become isotonic, and then they don't have to worry about, regulating osmotic pressure. So, humans, as, we have, as multicellular organism, that's basically our kidney. What our kidney is doing is kind of regulating water in our body. Okay. All right. So we just talk about facilitated diffusion, and that include things like aquaporing, for example. And it is totally a passive transport because you don't really need energy to put in. It literally goes from the high concentration to low concentration. And so this goes back to one of the earlier questions. So when it comes to physiology, for example, you think about the liquidy compartment in our body. There's the intracellular fluid, right? So that's every cell's cytoplasm. And then outside of the cell, there's also, within tissue, there's interstitial fluid. And then there's also your blood plasma. So the reason we are talking about diffusion and facilitated transport and active transport is because things are crossing these walls all the time. So if you think about the plasma, so your blood carries oxygen, and when the oxygen is delivered to the tissue level, oxygen will basically diffuse across the capillary wall and then goes into the interstitium and then goes into the cell so that the cells can use the oxygen. But then other things like, glucose, for example, will actually need a transport protein to help them get across the membrane to go into the cell. So the reason we spent so much time trying to understand membrane and all these transport mechanisms is because you really do need to understand these mechanisms so that you can understand physiology. Interstitial fluid, it's pretty much the same as extracellular. I mean, extracellular means outside of the cell. So if you look at this picture over here, they're actually talking about interstitial and the plasma both as being extracellular fluid. But interstitial is literally just the fluid that's being trapped in the tissue, and it's not within the blood vessel. Does that make sense? Okay. So so that's so all the ones that we just talked about, so diffusion, osmosis, facilitated transport, so those are all passive transport. Active transport literally means you are transporting things from low concentration to high concentration. So, whatever you're transporting is going against its concentration gradient. And then because of that, you actually need to put in energy. So the main difference is you have to use energy. So a classic example, of course, is the sodium potassium pump. So guys, every time you see the word pump, that instantly imply that it is an active transport. And the sodium this particular pump will basically use the energy from ATP hydrolysis. And then at every turn, it can actually pump 3 sodium outside of the cell membrane and then pump 2 potassium inside the cell membrane. And for most cells, the reason it is doing that is actually it uses it to establish what we call membrane potential. So because these ions are not evenly distributed across the membrane, so if you look at these pictures, on the left, it's the extracellular fluid, and on the other side, it's the cytoplasm. So sodium ion is actually already very high concentration on the outside, and then very low concentration in the cytoplasm side. And then the pump is actually using a lot of ATP to pump additional sodium outside of the cell. If you look at this table over here, it actually tells you, so inside the cell, the sodium concentration is about somewhere between 5 to 15 millimolar. And then outside of the cell is 145. So it's actually a really steep concentration gradient. And then the cell is actually using more ATP to pump sodium to the outside to maintain that steep gradient. And it's a chemical gradient, but it's also an electric gradient because sodium actually carries a positive charge. So chloride also play a role over here. So, when chloride gets pumped unevenly, it carries negative charge. Potassium, of course, also carry positive charge. So the combination of a lot of these ions, their uneven distribution across the membrane, basically give us the membrane potential. So on the two sides of the membrane, the voltage is not the same. And so potassium, if you look at it, it's the opposite of sodium. So it's really high inside the cell, and then it's very low outside the cell. And the pump is actually also pumping it against its concentration gradient. So always bring in potassium from outside to inside. So the electrochemical gradient, so, like I said, it is a chemical gradient, but it's also an electric gradient. And another example of these electrochemical gradient is also, there's these proton pump. So they literally will use the energy from ATP to pump hydrogen ion, which is also called protons, from low concentration to high concentration. So, this is actually a picture showing the lysosome. So lysosome contains a lot of enzymes that can degrade things. And also the pH inside the lysosome is actually very low. So inside the lysosome, there's actually a lot more proton than outside. And the cells will actually use the energy provided from ATP to pump even more hydrogen ion to maintain that acidity inside the lysosome. And the next lab we're doing is cellular respiration. So the electron transport chain, kind of rely on the hydrogen pump. So I'll talk more about it a little bit later. So when it comes to active transport, if it's just carrying one thing, either an ion or a molecule, it's called uniporter. But sometimes this carrier can actually carry 2 different things. So if those 2 things are traveling in the same direction, it's called symporter. But if those 2 things are traveling basically on opposite direction, that's called an antiporter. And active transport can actually be divided, depends on where the energy comes from, to be divided into primary versus secondary active. So primary just means that the energy comes from ATP. So the sodium potassium pump, again, it uses the energy that come from ATP hydrolysis, and then it pump, basically sodium outside the cell, and then it brings potassium inside the cell. And actually, if you look at this number, so in some cells, not actually most cells actually use about 30% of the ATP to basically allow this pump to function. And if you think about ATP, that's the cellular energy currency, right? So it's cell money, it's cash. So if you're spending 30% of your household income to maintain something, that something has better be really important. So that could be a car payment or your mortgage. But whatever that thing is, if you're spending 30% of your household income to try to maintain it, it has to be really important in your life. So the sodium potassium pump is really important for the cell, and that's why it's using so much energy to make sure that it is functioning properly. So secondary active transport, which are also called co-transport. So basically, you're not directly using energy coming from ATP, but you're actually using another concentration gradient to kind of fuel, to provide the energy. So an example of this is actually there is a sodium glucose syn port. So if you look at this transport protein, it can actually bind to both glucose and sodium. And remember, there's always a lot more sodium on the outside of the cell than inside the cell. So there's a very steep sodium concentration gradient. So this transporter will actually bind to the sodium and then the glucose. And because the sodium transport is energetically favorable, so it can actually use that energy to literally transport glucose across the membrane. It's simple because the sodium and glucose are traveling in the same direction. So both of them are going into the cell. And it's secondary because the energy does not come from ATP directly, it comes from the sodium gradient that we've been using the pump to establish. So this is one of the reasons why the cell used that much ATP to establish this gradient, because the cell can turn around and use the gradient to do work. And we just talked about the proton pump. Same thing, it can actually set up a pretty steep proton gradient across the membrane. So in animal cells, a lot of time we do use the sodium gradient for doing work. In plant cells, they actually use the proton gradient for transporting a lot of things too. And then we talk about lysosome. So there is actually a proton pump across the lysosome. In plant cells, they have the central vacuole that also has proton pump over here, and can actually help them maintain the pH. Okay, so this question says, how does the pump make the interior of the cell more negatively charged? And the correct answer we're looking for is actually because in every turn, it will pump 3 sodium outside of the cell, and then it will bring 2 potassium inside the cell. So, over time, it will actually, the outside become more positively charged, and then the inside become more negative charged. The pump does not transport anions, so it's literally dealing with only sodium and potassium. Both of those are cations. And if it's equal number, then it's not going to be able to generate a membrane potential. Malika asked the question about the sodium. So the sodium pump definitely is pumping sodium outside the cell, and that's against concentration gradient. So that's definitely active. The sodium glucose syn port. So that one, if you look at it from the sodium's perspective of the sodium ion, it is passive because the sodium is flowing from high concentration to low concentration. But that carrier process is still considered active because you're actually using the the energy released from sodium flowing in to fuel the transport of glucose. So it is a little bit more nuanced than that. Sometimes you can't just say active versus passive. It kind of depends on who you're talking to, so who you're talking about. Okay. So this one is a little bit tricky. So it's basically saying in which situation would passive transport not use a transport protein. If you look at every single one kind of case by case basis, if you look at water flowing into a hypertonic, remember water follows salt. So this is energetically favorable, but by simple diffusion, it happens too slow. So that's why we rely on aquaporin for most cells. So that one does use transport protein. And then glucose being absorbed from the blood. So glucose cannot cross the membrane on its own. So it definitely rely on some kind of transport protein or channel. And then ion, same thing. Ion is too charged to cross the membrane, so it doesn't even do diffusion. It definitely rely on channels. So the correct answer we're looking for is actually oxygen, because oxygen usually just really, it is moving across the membrane by diffusion. And we don't really have the membrane protein to help oxygen move, because they don't really need that help. Usually it's just the oxygen concentration gradient in the lungs or in your tissue. Okay, so we have Bahar, Jamie, Will, Anina, and Nikhil on the leaderboard. Okay, so far all these transport we're talking about are very specific molecules, or sometimes they bind to two different molecules. Bulk transport basically means that you can move actually a whole bunch of stuff into the cell or outside of the cell instantly. So first of all, bulk transport is also an active process, so you definitely need ATP to fuel them. And then depends on the direction. So it can either be endocytosis, where the cell can swallow something, or exocytosis, where the cell will secrete something. And depends on what the cell is swallowing. So if the cell is swallowing a really large, a whole cell, for example, so if this is your immune cell and encounter bacteria, your immune cell goes through phagocytosis, and phagocytosis literally means cell eating. And then the bacteria goes into the cell as the phagosome fused with the lysosome, and the enzyme will basically digest the bacteria into pieces. Other example of phagocytosis, amoeba, for example, they can feed on paramecium, and then paramecium can feed on bacteria. So, in the natural world, if you're bigger, you eat the smaller guy, and then, the smaller guy can eat somebody that's even smaller. But, all these feeding are also through phagocytosis. And then the vesicle goes in for amoeba and paramecium as food vacuole, and then they also fuse with lysosome, and then the content gets digested. And another form of endocytosis is pineocytosis. So pineocytosis literally means cell drinking, and it's actually the cell usually, instead of taking in a large whole cell or large solid molecules, it actually taking fluid. So it's a little bit different because it also forms these vesicles, but these vesicles are smaller. And also these vesicles don't really fuse with lysosome because, they just contain fluid. So they might be used in other ways. So some example of pineocytosis, if you, think about your small intestine, these epithelial cells have a lot of microvilli to increase the surface area. And they actually use pinocytosis to absorb nutrients. So, maybe, small lipid and stuff like that. And then exocytosis is basically, the cell will secrete. And, a perfect example will be a lot of neurons secrete neurotransmitters, and also endocrine cells like pancreatic beta cells, for example, they secrete hormones like insulin. So they actually contain these, granules or vesicles that contain the neurotransmitter, and then the vesicle will fuse with the cell membrane, and then the neurotransmitter gets released into the extracellular space. And that's how neurotransmitters basically secrete things like dopamine or serotonin, or insulin and stuff like that. Okay, so this one is just a definition question. It says which transport mechanism can bring whole cell into a cell, and the answer we're looking for, of course, is phagocytosis. Whoa, okay. So this one says, what happens to the membrane of a vesicle after exocytosis? So if you look at the picture, so we have a secretory vesicle that contains neurotransmitter. So let's say it contains dopamine, and then the vesicle fused with the membrane. And so who leaves the cell? It's the dopamine, right? So the neurotransmitter leaves the cell. The membrane itself, because it's made of phospholipids, basically it will become part of the plasma membrane. So we have 22 people giving the correct answer, which is actually literally, so this is why intracellular trafficking works, because everything is made of phospholipids. So when they encounter other phospholipids, they can actually fuse with each other, and then they literally become a part of the plasma membrane. So let's see who's on the, that's actually my last slide, I think. We have Will, Jamie, and Bahar on the podium. And then Anita and Nikhil are on the leaderboard. So good job, guys. And so there are a couple of you guys who asked me about pre-lab questions about osmosis or hypertonic versus hypotonic. Did that answer your questions, guys? Okay. If not, you can also, later on when we break out, you can also ask me. So I also, before I send you guys into breakout room, we kind of talk about lab report. So it is coming up. So I have opened the lab report module. If you go into Canvas and go into module view, it's right under the getting started module. And so basically, if you click on the guideline, so highly recommend everybody to download the guideline. It kind of just talks about the structure of the lab report. Oh no, I wish I had kind of, that's okay. I think I can show you guys. over here, let me open up this one too, just so I can show you very quickly, because people were getting a little bit anxious about the lab report, and it does carry, it does carry quite a few points. So you do want to do a good job with the lab report. It takes forever to load. Okay, so if you just download the guide, it's a Word document. It just talks about the different parts of the lab report. But usually, you want to have a title. So it kind of is the format like a paper. So you have a title, and you want to have an abstract. People usually, when you write a paper, the abstract is the last one to write. Because you want to write all the other parts of the paper so that you know the full story, and then the abstract is it's the summary of what the story is all about. So you should have an introduction, so talk about some background information. I actually kind of like this graphic. So the introduction, the structure is usually going from general to specific. So when you're writing a paper, you want to introduce the background, and maybe even talk about what other studies have found and stuff. And then the last part of the introduction, you want to talk about, this is the transition going into your own study. So you want to talk about your own hypothesis going into, so why are we interested in, remember, hypothesis is your tentative answer to your question. So why are you interested in that particular question? What is your hypothesis? So the introduction is going from very general, the large topic, large picture, to very specific. So, this is what I'm gonna talk about in my report. There's usually a material method one to kind of just talk about how the experiment is done. So you want to talk about the experimental design, and then a little bit more specific of how the experiment was done. And then there's the result. So this is the part where you, if you have figure or or data and stuff, so you present and you describe your data and stuff. And then in the end, you have a discussion. And discussion is, so in addition to just presenting the data, you want to really interpret the data. So what does it mean in basically from a large picture or big picture viewpoint? And so this is where you want to talk about, your result. Remember, we started with a hypothesis. Did your result support your hypothesis? Did they falsify your hypothesis? And how does it compare with other studies, right? And are there most experiments, if you're working in the lab, most experiments fail. So are there some possible errors, or what are the sources of errors and stuff like that? And then what's your general conclusion? And then of course, this being a science lab, you need to give me at least 4 references. And Purdue OWL is actually a really good resource if you guys want to learn more about citation, or just the format of citation, or in general, what are some good practice in terms of references. And in terms of the point distribution, so basically this what the point distribution is. And so when you do go in and turn in your lab report, so this is what is, the assignment kind of looks like. But the report also has a rubric attached to it. So before you guys write your report, before you submit your report, and by the way, the report is due on October 18th. So we still, we do the lab next week, and then you guys have, I think, 2 weeks from there to do to write the report. But this is what the kind of a criteria is. So abstract itself is 6 point, but just read the description for the excellent part, right? So that's kind of what you're striving for. And the introduction, so make sure you have all the different pieces. So as long as you have all the different pieces, and then there are not a lot of errors or typos and stuff like that, people usually do pretty well with the lab report. So definitely read the rubric and read the guideline. So the rough draft, okay. And Nikhil says, so let me think about the rough draft one. Yeah, so we're all, because everybody's doing the same simulation. So this lab is based on the cellular respiration simulation. So everybody will be basically trying the answer. So, go through the simulation, see what they're trying to answer, what question you're trying to answer. But yeah, in terms of the question you're trying to answer, yes, everybody is. And yeah, so the lab report is the lab that we're doing next week. And one more thing. So in addition to all the things we said, all the different parts, results, discussion, and stuff, there's also this one category about creativity. So this is where things like typos or grammar errors, you can actually get great detection, and also make sure your language is fairly concise, and make sure there are no errors and stuff. And in general, if people use more than 4, so 4 references is required. If people use more than 4 references, I do have a tendency to give some extra credit. Yeah, so that's the lab report. I wasn't planning on you guys submitting a draft, but for the presentation, I actually do have planned for you guys to submit a draft, and then I actually will send it out to other people for peer review. So it's a more extensive process, and so why don't we, let me think about it. And then also, if you guys, when you do your data reflection today, go ahead and put it in there and just see if you have a preference. Do you prefer to submit a draft, so that you have an opportunity to get some feedback from somebody, and then modify it or not. It's a it's doable, but it's a more involved process. I wasn't planning on it, but I'm not 100% against it either. So I can definitely see the value in that. So yeah, go ahead.
So I guess I'm trying to understand. So we're doing the lab report next week. So do we just do the simulation and we just go find articles about the topic?
Yeah.
And then write the literature review basically. And how are we doing the methods and result? I guess I'm not understanding that part.
So whatever you're doing in the simulation, that's your experiment, right?
Oh, okay.
Yeah, so the material and method, basically that's also taken from the simulation. It's been a while since I did the simulation, but yeah, I think you're using some instrument to measure oxygen, for example, and there are other machinery and stuff involved. So how exactly did you measure cellular respiration? That's what goes into.
Okay.
Yeah. So if most people agree on a draft, I'll think about it. So then we can actually we can do it. Let me think about. I might redistribute some of the points, but yeah, but the lab is next week, and at the moment the due date is October 18th. Yes. Okay. Yeah. All right. So any other question? And guys, if you want to see your quiz, you can just stay behind, and then I can show you guys your quiz. But let's do the class discussion first. And I'm gonna publish, just like every other time, I'm gonna publish this homework thing, which feels a little bit silly. I just don't really want people to do it before we have a chance to go into the breakout room. And we are going into 8th breakout room. Actually 9 breakout room. We'll make sure that we have 3 to 4 participants per room. So I think, yeah, Kay, you can stop recording it, I guess. From now on, it's just logistic.
Sorry,